thanks everyone for coming. Wow, it's I think I say finally we're ramping up, probably because we have also a very popular speaker, and it's great to see you here for this other session of the Machine Learning Coffee Seminar. We have here with us Professor Ville Mustonen, Professor of Bioinformatics at the University of Helsinki, and well, he's also affiliated with, you can see here, a lot of other institutes like the Center of Excellence in Antimicrobial Instance Research. And he's going to tell us about his research. And thanks, Ville, for coming.
Thank you, Luci, for the invitation, and it's good to be here. So I last time in this series I presented about our cancer mutation work. That was 3 years ago or so, and today I will talk about microbial growth. I tell you a little bit first about our group. So the group is called Bioinformatics and Evolution. We have at the moment 2 postdocs, 4 doctoral students, and 4 research assistants. There's a good representation in this corner table of our group members, and I'll introduce them later once we get to the end, because the work that I'm going to talk about today is done within the group as a joint effort. And we are biologically interested about evolution and evolutionary dynamics, and especially in the context of trying to understand drug resistance. And we approach this problem from 3 different viewpoints. We work together with our collaborators, essentially to produce empirical and experimental data. And so that's kind of a big component. Then my group's core competence is to develop methods to make sense of this data. We have often solid evolutionary and ecological theory grounding for the methods. And at times, we also develop kind of more algorithmic things as needed to work with this biological big data. But I'd say the focus is really on these axes here. And we do this more when it's necessary. We are very happy to use tools that other people have done if they are sufficient for our purposes. And we are based in 2 campuses, in Viikki campus, here in the Organismal and Evolutionary Biology Research Program, and then Institute of Biotechnology, and here in Kumpula campus at Department of Computer Science. Okay. So antimicrobial resistance, you have heard about that most likely, is becoming an increasingly serious global health threat. And these 2 articles are from last spring, and they are in fact highlighting that the antimicrobial resistance is now a leading cause of death worldwide. So they were basically popular press articles about this particular systematic analysis study that was published in Lancet earlier this year. So this is becoming a large problem, and it's kind of, we have had this COVID pandemic, which was very quickly and proceeding thing, and antimicrobial resistance has a different dynamics. So it's progressing, and it's a pandemic, but it's much slower. So then it's somewhat easier to ignore. But I guess this phase of not caring about it is over now, and people are putting efforts and understanding it more, and it's kind of commonly accepted that it's a problem. And antimicrobial resistance, in its simplicity, you can think of it that you have some antimicrobial that works initially effectively against the pathogen, but then the pathogen acquires some genetic elements, let's say, and after that, the antimicrobial doesn't work. So that's antimicrobial resistance. So then you have lost one way of controlling a type of infection. So antimicrobial resistance is complicated. And the reason for it that, let's say, the spatial aspects, they go different across different scales. So environments affect the AMR risk, and how hosts are behaving in the environments, what are their activities there affect AMR, And then if you go even to smaller scale, we can see, for instance, here human gut, and there are microbial communities there, and within these microbial communities, the community structure, what species are there, what are their genetic compositions, affect antimicrobial resistance risk on how it's developing. And this is kind of a pictorial thing. You have low risk components, high risk components. Let's say you have clean water versus you don't have clean water. You have good farming practices. You don't have good farming practices. You have clean hospitals, which, you have crowded hospitals, which are probably not so clean. And then you have clean hospitals with space and sanitation. And also aspects such as how interlinked we are these days, so that the antimicrobial strains, if they arise somewhere around the globe, they will find their way to everywhere, essentially. So the topology has changed in that sense because of interconnectedness. And also, like I said, these kind of intrinsic eco-evolutionary processes affect. So if you have certain type of communities, they are harder to invade by pathogenic strains, whereas if you have other type of communities. So this is a big picture and complicated, and for that reason, we applied for the Finnish Academy of Finland Center of Excellence funding a year ago, and we got funding for our Multidisciplinary Center of Excellence in antimicrobial resistance research. And we are now building up our activities. We have 5 PIs in this project. Anu Kantele from Meilahti is an infectious disease doctor. Then Marko Virta is a professor of microbiology, and Anu is professor of infectious disease. So Marko is doing environmental microbiology, so collecting samples, analyzing them like wastewater and so forth. Then we have Teppo Hiltunen from University of Turku. Teppo does very specific experiments with microbial communities in the laboratory. So you can control lots of the complications by doing that type of experiments. Then we have Salla Sariola and her group from the humanities, social sciences, trying to understand how human activities affect the antimicrobial resistance dynamics. And then we have my group where we try to kind of do integrative analysis from theoretical viewpoint and also using, let's say, bioinformatics methods on this type of data that we are collecting. So we need this joint effort where we have groups across disciplines because of the problem which is this complicated. So today I will focus on one aspect, namely microbial growth. And you can think about microbial growth, or at least I think about it in a way that it is the killer app. It's really essential to understand how therapies work, be they antimicrobial therapies or be they cancer therapies. It's essential in biotechnology applications. And it's also essential to understand ecological systems. So growth and its quantitative understanding, and at some point its controlled manipulation, are kind of key enabling technologies. So that's why we want to understand growth quantitatively. Okay. So this is a project we are doing together with Jonas Varinger's lab from Gothenburg. And Jonas has been developing these kind of high density systems to measure thousands of colonies at one go, or in a parallel way. So what you have here is a plate which has a solid plate. It doesn't have any wells, but you can plate colonies, microbial colonies there. And one plate has 1,536 colonies. And then you can let them grow on that growth substrate, and you can do scanning of images while they're growing. So then you have an image time series, which you can convert to estimates of the population growth, what you see here in the on the lower panel. So you can see the optical density here as a function of time. So initially very little happens, but then you start really growing, and then the growth plateaus at some point. And here you see a typical setup when you study some set of microbial colonies. You do typically a control measurement, so that's the environment, certain nutrients, and then do your measurement in some compound of interest, here antimicrobial hydroxyurea. And you want to understand what are defining the growth there. Let's say the genetic elements that are giving you that type of growth behavior. So that's the basic setting. And the technology is really nice in the sense that it allows you to do very quickly phenotyping of thousands of different populations. But growth is difficult to understand. And here is like one aspect of that difficulty. So the data set I'm going to show you and its analysis today consists of 4 different plates, 4 different conditions, and all the 1,536 colonies are of the same genotype. So it means they are the same strain. So we kind of remove the genotypic variability, but in a different usage of this platform, we typically would have lots of different genotypes because we are also interested how the different genotypes are affecting growth under the conditions that we are studying. And I'm showing you here the mean growth rate, growth curves of these colonies. You can see that in some condition, the mean growth starts slower, doesn't go to so large colonies, whereas in others, it might start quicker and go higher. And these are kind of some deformed logistic growth models, right? They are not exactly the symmetric S-curve that you know from your studies, or like you have seen for sure, but you can imagine that you have some ways to distort that, and they look like that. So the lag time, typically it's called the time when it really starts to grow, can be longer or shorter. And then when the stationary phase is reached, you can see where you plateau here. That would be called the yield of the population. But this mean actually is masking quite a bit of variability. So I'm showing here you now the mean for one plate and plotting all the 1,536 individual curves there. So you can see that actually, although they have the same genotype, those populations, there is a lot of variability that we would want to explain, especially for the reason that if we want to utilize this type of growth data across different genotypes and use all of the information, we would want to do that and understand what are the sources for this variability. We have used this platform in the standard mode where you use every kind of 4th colony is used as a control position, and you can normalize by the control position. So that's something that has been done. But then, of course, you lose quite a bit of measurement capacity for the controls, and you tend to use only some very kind of summary statistics of these shapes. And we want to get rid of that. We want to use the whole growth law of these populations. Okay, so that's the setting. And we look at this data typically not as the whole population growth curve, but we look at it in terms of the relative growth rate for population. And it's nothing more than just the derivative of the growth curve, and it's per capita at that point. So we have this delta n per n in our y-axis. And what you can see here, for instance, is that now the mean growth looks like that. And you can see that it peaks. The highest rate, relative growth rate happens at this time, and it's this value, and then it starts to decay. And you can see that there's then one individual colony, and you can see it's quite different from the mean. Okay, so let's start to analyze the data and try to understand the different components of growth. So here's just a different visualization of the stochasticity there. And sorry, I didn't have time to do this before the talk. So the you can see how the variance of this mean growth rate looks across the different plates. And with this, we actually go and define our baseline expectation. So let's call it the null model for growth. And the null model is that our model for rho is just going to be some time-dependent function. So we go to every time point and we basically take the mean value, because our metric for error is just the least squares error. And you can see here we have these different error profiles as a function of time. So this is one way to look at that. And this is already quite telling, because if we look at this now and we look at the per colony location error spatially, you can see that that error, so how is the individual colony and its growth rate differing from the plate mean, has clearly some spatial structure, right? So you can see that there is, this is not across the space randomly distributed. And this is now something that we essentially would want to break down to components, what's causing this, and then perhaps get rid of that to like utilize the whole growth curve data rather than just partial aspects with local control positions correcting those partial aspects. Okay. So we first built intuition by just doing black box machine learning analysis. So we want to now express this growth, and now you have see the sub-index I, so it means that now we are building a model for each one of the colonies in this plate, and we say some function of the colony location being row and column, and the actual population density there. So we want to use the population density at that time to make a predictor. And what you can see from here is that you can build a decent predictor for the training data for the growth using these components. And with the test data, it also performs well. And there's like small variability depending on how we filter so you can do slightly better than this if you filter a bit more the growth data, but qualitatively the pattern is the same. Namely, the pattern is that the very initial part of the growth seems to be fitable, but it doesn't generalize. And then you have lots of value by knowing what's your state in the growth, like how large the colony is. And then later in the experiment, it starts to matter what is the position that you are in the plate. So this is the random forest regression analyzing this data and looking at the feature importances, building us intuition what components our model should contain. Let's look if the actual raw data looks consistent with that. For the spatial effect, it looks clearly to be the case. So if you have a look of these for one plate and you look at time slice 50, 100, 150, 270, remember these are all the same strains, you can see that actually the boundary layers are definitely growing faster than the interior ones. So there's definitely a visible signal there that you can take into account. That's something we will build into the model. But let's first focus on the observation that the very initial phase looks somewhat unpredictable. It looked fitable, but not predictable. And if you actually go and read about growth from biology textbooks and articles, this kind of makes sense, because you can think that even though the genetic part is the same for a cell population, it is in some physiological state, which is reflecting, let's say, the pre-culture conditions. And it will take some time before the cell population starts to grow in the new environment in a let's say, the way it can grow there. So there's this initial thing. And this is modeled in the literature using an adjustment function. Parana and Roberts are behind it. And it's basically alpha of I. And it takes into this initial physiological state, reaches a maximum R0, and you leave the initial physiological state with some rate M. And this is now nothing but a reverse logistic. So you start from something small and you go to R0. And importantly for our experiment here, the initial physiological state is a local parameter. So that's a degree of freedom we allow for every colony. But then the two other ones are properties of the genotype of the strain. So the rate it takes to adjust to the new environment, M, and then the maximum growth rate on that environment. So we have added now one local parameter to the model, and then two global parameters. So not a lot. And now let's see how that affects the fits. So you can see that this model M1, where we express this one and then keep the rest of the fit as the mean population growth rate, you can see that you are, for instance, plate 1, you are getting rid of this first peak, and you are generally improving the fits here in the initial points, as you should, because that's the part which we are trying to model with this M1. We take the initial adjustment into account. Okay, but let's see, the spatial error looks very much like before. So the improvement that we got does not remove at least a lot of the spatial error. There's some improvement, but very little. So it looks very similar. So we need to make our model a little bit more complicated. And as we have seen with the machine learning analysis, the space does matter. So what we are doing now is that we'll try to express this growth rate in one colony location as the initial adjustment function, and then some uniform field, global field that depends on the location of that, but only in the sense that how far it's from the boundaries. And this is, of course, a function that we don't know, but we are going to learn, and that's learned globally now, because that's the same function for the whole plate. And if we do that, you can see that we are increasing and getting even better. So you can see the green line here. It's starting to remove some of this part of the error. Same here, and same here. So this is clearly improving, and it's already getting rid of some of the spatial aspects pretty nicely. That's nice, of course, because now we have included some components of space. So you can see, for instance, here, there is spatial aspect in the error. Some of that is definitely moved. There is spatial aspect that's moved. And here, perhaps even more so. And clearly we are getting to the right direction. But we haven't yet, if you go back to the machine learning fits, the spatial aspect is actually quite important, but equally important for sure is the actual population size that the colony is in. So that will need to factor in next, that we get this model to take that into account. So level number 3, we are then expressing this dependence on the population size as some unknown function phi of the population density. And the issue is, of course, that if it's an unknown function, we would need to learn it somehow. And we use here a particular form, which I will justify in a moment, for basically self-consistently tying up with the other parts of the growth rate to the population size. And if we do this type of a model, what we can show is that now it starts to perform extremely well. So that's the red line here. So you can see that you are essentially explaining all the errors away here. Not quite all, but a lot. So this is substantially improved model. And if you look at the spatial effects, you are now almost free of them if you go from the null model to here. So you have removed vast majority of the spatial bias by having this model, which takes into account the size where the population is at given time point and the spatial location of it. So this is kind of nice, and this would be already a good endpoint in a way. So we have almost model-free, effective factorization of the determinants of growth. But for several reasons, this is not quite satisfactory. First of all, we did actually need to do kind of lot of learning from the literature to have kind of the right parameterization. I come back to that. So that we had this alpha function and so forth. Then this is the really difficult issue. Epsilon is now a time-dependent field that we learn. So that's slightly difficult because if we have a plate where we have lots of different genotypes, obviously we can't learn a field for every one of them. So then actually we just get our own data back. So we are overfitting. So this is basically not satisfactory, albeit works. So we can do that. And then the third point is that, well, where did we get this dependence on phi? So there's some function. And there also have to say that if you just try any function of n, it doesn't really help at all. So that was already quite involved one. And there's obviously a reason why it looked like that. So let's step back a little bit and start from the beginning. So in fact, this is now where we are. So the way to think about this is that because the plate is just a solid plate and the colonies are growing there, when they are growing, they are consuming nutrients, which we don't observe, but they are actually impacting the other colonies' growth via consuming those nutrients. So they are, if you grow very fast, you are eating more than your neighbors are eating. So what we can say that there is a latent variable, some growth limiting nutrient concentration, and it has some dynamics, which is spatially coupled by a diffusion. So the S has a diffusion to its neighbor. So this is a mean concentration at the location I, and you if the mean concentration is higher, then this location I concentration gets a boost via diffusion. Otherwise, you lose to the environment. And then the colony is actually consuming the nutrients there with some function that we don't know what it is. And then actually the per capita growth rate is going to be expressed as alpha I as previously, but now everything else is just put inside this function of the nutrient concentration, which we also don't know. So now this is the tricky part here. We have two couplings functions that we don't know, and we don't observe this latent space. This is something that we struggled a lot to get it practically to work. And then again, just reading a little bit more, we thought that we need to kind of fix something. So we fixed, based on the literature, the substrate consumption function to be linear in the derivative of the growth or the population size, and linear in the actual population size. And the interpretation is that one is nutrients used for growth, and one term is the nutrients used for maintenance. So this is now fixed. And while we keep the actual from concentration to growth rate as a very generic monotonic function. And this functional form is just a monotonic function. I think it's not biologically so important what this is precisely. It's more actually, you can, once you have the fits, you can check how this looks, and that tells you the biology, and not the way we just parameterize that. So that's not relevant. So it's, think about it, any non-monotonic function, which is kind of genuinely flexible. And now if you plug these in, then what happens, this is just from literature showing you what people have done. So this is from the literature set of what I'd say are just non-monotonic, monotonic functions. So we probably cover most of them just with parameterizers we have, at least many of them. And here are the couplings to the substrate. And here you can see actually it's all done just on the growth and not on the maintenance. But we do need the maintenance when we actually go to the data. It's very important there, the maintenance term. And if we do that, we have in fact extremely good fits across all the plates. And now this is a very low parametric model, because we don't learn some epsilon time-dependent field, time and position-dependent field, but via going this latent space kind of data augmentation, we have two global parameters plus the diffusion constant. And then we still needed some way of doing this nonlinear coupling here. But we are talking about a handful of parameters for a data set that has 218 time points times 1,536. So it's really expressive model for the data. And you can see that it does extremely well. It's the best model here, albeit it's much simpler than this one, and kind of removes the spatial bias there. And like I said, what you can get here, you shouldn't stare at the kind of parameters that we put to this coupling of concentration to growth. I don't think they are very relevant. It's just the shape actually tells you what's happening biologically. And there you can see, for instance, that in these different experiments in conditions, how is the growth rate modified when the concentration drops. So all these experiments actually start at levels of nutrients with this interpretation where the growth is not yet nutrient limited, but then they all end at a point where they are nutrient limited. So they could be growing faster if you had more nutrients. And then we get also biologically out the maintenance, like how costly it's to maintain versus how cost is to divide or make new cells. And there's roughly a factor of 100 there. And then we get the diffusion constants. Okay, so to summarize, if we have a model which is explicit in modeling the environment, it works extremely well, has only very few parameters, and compared to models where we try to do the spatial effect just by raw inference, so that we have this latent space model for the how the interaction is mediated. And this is now for us very exciting for the reason that the way we have been using Jonas's platform is in a way that we have wanted to correct always the spatial aspect by using these control positions. And that has been good for the studies we have been using it. But now if we understand at this level, how is the spatial effect generated, actually, I could say that these interactions between colonies become a feature rather than a bug to be corrected. So feature in the sense that they are actually super interesting. They are not technical artifacts. It's genuine biological thing. There are resources, populations are spatially competing for them, and it has consequences for growth. And with this platform, we have done evolutionary experiments where you grow colonies, you replate them, you grow again, you do this kind of transfer cycle for long times. And you can imagine that depending on your location on the plate and who are your neighbors, the kind of selective forces acting on you are going to be different. So I think there's going to be something very exciting coming from here. So part of our massively parallel eco-evolutionary experimentation. And what we are going to do here next, so we don't want to we want to make this automatic now, how to think about that type of latent space models. And this is something that would be very good to hear your opinions. So how to go about this type of problems when you have this type of structure, that there's a latent space, we know something about it. We know, for instance, that's mediated spatial effect, but we don't really know the shape of this, and we don't know the shape of this. So what are the kind of obvious techniques to use? So we resorted now here, fixing one based on the literature, fixing the other one in a rather general way, but it would be really cool to do this kind of automatically across some function sets that are sensible for biology. So that's something we can discuss. And this is in fact something that from my group, Karen, who is here, is going to do a master's thesis on this symbolic regression aspect of this problem. And Dovidas is working on a PhD project related to that. So I'll introduce you in a moment when everyone can turn around. So that's something I'd be very keen to hear your opinions. And other than that, I want to thank Florian, PhD student, who is the lead author for this paper, and then other group members who have been part of this project, and Academy Finland for funding this part of the project. And I think I went 5 minutes over the time, but we have still 10 minutes time for discussion. Thank you.
Really interesting stuff, and I understand correctly that you started with this place and you wanted to then predict how the growth develops in time, and it seemed that with this M3 equation you're kind of using some control-like aspects by taking the interval over time and using previous time states to be able to.
Yes.
In M4 it was almost like learning the rate of change of that. So really, that for me was a lot of control-like things, but I was a little bit confused about, which I think is fantastic, like a very nice way to go about it, but I'm a bit confused by what you mean by latent space and latent models. Like what exactly models are you referring to? And are these like generative models or?
Okay, so thank you for that. Let me say one thing. I think I missed this one. So the M3 functional form, we didn't get this M4 level to work for a long time because we didn't have conceptually this nice separation. Like we were kind of moving both parts and we didn't really get to it. That's why I'm saying also that symbolic regression is so important, that it would do it for you automatically, rather than you always implement everything and then you have to be sure that you did that correctly and it's not working. Or you just didn't do it correctly and you don't know if it's working. So these type of things. So we don't want to do that anymore. But when we did this one, M3 already took in some of the theory. And the theory is such that if you have a reduced model here, namely, this coupling to the population density is just with this growth rate, so the derivative. And this is a linear function. Then you can solve this analytically. You can integrate out the latent space. And then you have a phi function, which is giving you this functional form.
Right. Yeah.
So that's the kind of a case that you can do analytically. And we can even do that with the diffusion. But importantly, now let's go back to your question. So this is now the latent space. We don't observe it. We don't have data to fit it. Right? That's what I mean. It's genuinely latent. We know that there is such a thing as the environment, and these cells have to use some nutrients from it. And the way the fits go at the moment, or this whole inference, you have a model here. You choose something here, and then you can always integrate this latent space out and put it here. So we learn it as we go, and we always forward integrate. There are some cases, depending on F and depending on this coupling to S, which you can solve analytically. But you can see, because the spatial coupling of the latent space is here, you can't really do that as a rule. So now I guess my question to the audience is that we have looked a little bit and we have seen this type of latent space augmentation for physical models, for instance. So the idea is here that we know there's a kind of generative model in the latent space which we don't know, but using that problem we are trying to do looks much simpler. So this is what it is, and we have basically been able to crack this because we know relative amount of biology and we know biophysics, let's say this kind of stuff, but we want to do this automatically going forward. So like, what would be the right way? It's some iterative way of solving it.
Like, it's tricky, but one, you kind of knew that you found the equation.
Yes.
And when you thought it was something that, like, you want to learn a Latin variable model, and then you want to like learn the dynamics of the system, but if you don't have the equation, I think a lot of these physics informed Latin variable models, they use Hamiltonian, so they have very nice equations.
Yeah, so they have a table of sensible equations, but I think we have that for biology as well.
Yeah, so then it's fine. You can, then you should have like.
Then it should be somehow like your function set is this, and then you would do automatic search there.
Yeah, there's some reason that's like doing also about cell biology. Could maybe find with find some reference for you. But yeah, people are doing definitely this stuff. But what exactly for your model were you using as a Latin variable model?
This is
just learned as
this is the equation that we have linear in this operator, and this we didn't like I said, we didn't make progress when we allowed this one and this one to kind of simultaneously change. And it's these are both terms that are somehow backed from the literature, so they are sensible. But actually the literature is very interesting because they have there chosen already kind of parameterizations for these functions that I think are arbitrary by fiat. They have said that it's some, and then they learn the power. But I don't see necessarily any reason why it should be like that. So it's just that it fits the data in that sense. And the data that they have might be much more kind of limited than I've discussed today. Because actually, again, I think the fact that the spatial concentration of the nutrient effects, I have a kind of intuitive feeling that it makes this model easier for us to learn, because we see at different concentrations, which we wouldn't see without that interaction. So kind of my assertion is here, based on intuition, that we get better models for these couplings when we have this diffusion going on. And you wouldn't have that if you would have just 1536 non-interacting things. So that's also interesting, I think. So in that sense, also it's a feature, an asset. If you have a model which is accurate, then actually you are doing slightly different versions of the experiment many times. So that's bound to be valuable and not just exactly the same one.
How well would this model work if the colonies were not evenly spaced on the plate, like they were somehow random, so some of them have more nutrients available than
we haven't tested, obviously. If this is the right model family, it should really work the same, because I think that the diffusion part is kind of basic biophysics, right? So it really should be. And actually, I should say here, I forgot that, but it's kind of why this is, the effect is there so strong at the corners is that if you actually go to the plates, you see there empty space here. So we catch that with this model. So we don't have to have the most boundary layer, if you see what I mean. So that they do actually really show the fact that there's no one there. So then we would need to test your kind of thought experiment, but it should work if it's really kind of, if we capture the spatial thing as we say we capture.
Yeah, it just seems to me that the most of the error in the final model is still in the corners and the boundaries. So.
Let's see. I don't think so. Here perhaps, yes. There is still
the top right one also.
Yeah.
If I understand.
Yeah. Definitely. But this is already like extremely good. So now the question is, should we still try to quantify how much error there is left? What we have done is that we have done averaging of the error per layer, and then seeing that how much it still is a non-straight line, but it has some slope. And I think you are right, there's still a little bit. You definitely see on this side that the layer number 1 is off a little bit more. You see here. So at least my eye takes some gradient there. Yeah.
There's also just a quick comment. You said that some other reason you have power loss. So one of my courses in my doctoral studies was theoretical biophysics.
Yeah.
And it's actually when you have a physical system or biological or whatever, so then you have to have power loss. So it's not arbitrary. And basically it comes from that your equation should work in all dimensions, whether it's a meter or feet, and it's actually a very interesting topic. And as a result, it really restricts the shape of the equation.
Yeah, I know. So some of these are based on scaling. So for instance, that if you in a 3D liquid compared to a solid plate, the growth dynamic scale slightly different with n. But I'm not talking about a slightly different thing. It's related. So I'm talking about how the nutrient is being consumed by the colony and what's its independence. And there's no one result from the literature for sure. And that's why what I'm trying to say here, that it's good to just do it with symbolic regression, see what comes out, and see how that talks with the literature. Because there are things that are kind of preconceived, and some of them are correct, probably, but you don't necessarily know that. So I'd much happier to do it kind of model free in that sense, that the only model that we put in is this coupling. This makes just total sense. But I would really want to do this as a, let the data decide it for you. So that would be the the kind of challenge. And actually with Karen, we are, with Dovidas and Karen, what we are doing now is we are making synthetic data where we have different ways of making a spatial effect. And they would have very different ecological interpretation. And now the question is, can you figure it out automatically from the data? So that you can you invert that growth thing to a good understanding of the spatial effect and get the right interpretation or not? So that we have different ways to put the spatial effect in.
Okay, thanks. Thank you again.